It's my pleasure to introduce our Case Unite seminar speaker series today, who is Dr. Joana Cabral from University of Minho in Portugal. Joana did her undergraduate training in biomedical engineering in Lisbon and did her PhD in Barcelona under the supervision of Gustavo Deco. Since then, she spent a number of years in the psychiatry department at University of Oxford and is now back in Portugal at Minho University. And in terms of Joana's research, it's no understatement for me, at least, to say that Joana's one of some thinkers in this still pretty new field of whole brain modeling. She's got over 50 publications in this field over the last 10 years, during which time I believe she also found the time to publish 3 young children, which is doubly impressive. But her scientific contributions, they've spanned a number of areas, including brain lesions, neuromodulatory systems, neuroimaging data analysis, and the area that she's particularly well known for is her work on whole brain modeling of oscillatory network activity. She's done some really interesting work in that field. And I know from seeing her talk and seeing these things with others, she thinks very deeply about these questions, and she also has a real talent for presenting complex ideas very clearly. So very much looking forward to the talk today. So Joana, take it away.
Okay. Hello, good afternoon. So I'm going to share my screen. Let me see here. Okay, it's here. Start. So first of all, I'd like to thank the organizers for inviting me to share these ideas, indeed, like all these thoughts I've been giving into trying to understand brain function. And I'm particularly interested in understanding brain network dynamics. It's been my field of research, either from using computational models, but also to look at brain activity itself to see which features we can extract to inform computational models. Because one thing is to develop a model, an abstract model, but it needs to link to reality. And so in the last couple of years, let's say, or even more now, I've been leaving the computational modeling a bit on the side because I feel that we need to gain further understanding into what we can capture from brain activity to see if our theoretical predictions fit the reality. So I'm going to show a bit what I've been finding on the dynamic behavior of brain network states. So I'll start with an introduction to cover a bit of the evidence on brain network dynamics and what are the methodological approaches that we can use. And then I will explain a bit or show some results that we can detect altered network dynamics in neurological and psychiatric disorders, and how this can bring us insights into psychophysiology, which I like because in a way these networks are what coordinate our thoughts and behavior. So ultimately understanding how brain network dynamics, which rules govern brain network dynamics, can help us understand our mind or consciousness without trying to be too, without trying to remain scientifically correct, but getting a bit into the field of psychophysiology is one of my interests. So just as an introduction for the ones who are a bit outside the field of connectomics, let's say, so we can talk about structural connectivity when we know that 2 brain areas in our brain are connected by white matter fibers, so by projections of axons that connect these 2 brain areas. So this is called anatomical connectivity or structural connectivity. But then there is functional connectivity, which is characterized by any type of temporal similarity in the activity of 2 brain areas. So if we consider 2 brain areas, NNP, we can compute the correlation between the activity in these areas and infer a measure of functional connectivity. Correlation or any measure of temporal similarity, covariance. So having established this, so we can again have the activity recorded in the MRI scanner, which is the bold activity. It depends on the blood oxygenation level, let's say, and then we can compute a functional connectivity matrix. But then recently, in the last already, let's say, decades, people have been finding that functional connectivity is not static. So it's dynamic over time. So if we compute the similarity, let's say, between the activity between different brain areas, we find this is constantly moving over time. So we can compute a measure of instantaneous connectivity and then see how this evolves over time. And if I project this, so I'm going to go into details on the methodology, but what this is showing is that there is a constant reconfiguration of patterns of connectivity in the brain that is evolving over time. So this suggests that there's a complex network dynamics that is not captured when we use a static functional connectivity approach. So nowadays, most of the research in functional connectivity is moving towards this concept of dynamic functional connectivity. So one of the things that has been, why this is relevant to these functional networks, is that it has been reported that different brain subsystems, so these systems of different brain areas that integrate together information, activate even during rest. So they are not just activated, these patterns, when we are doing a specific task, but when we are lying quietly in the scanner, we can observe the spontaneous formation of these network patterns. And there has been a lot of literature. These networks have been reported consistently across large groups of subjects, different research groups, so over 1,000 subjects. So there is really a consistency here. There are these networks of intrinsic connectivity or resting state networks that seem to integrate information at the system level. So the idea, and what is more important, is that these networks seem to be disrupted in many neurological and psychiatric disorders. So there is this concept now of pathoconnectomics or pathological connectivity, which I think motivates essentially why it is so important to understand brain network dynamics, because in a way these networks are coordinating something that may be relevant for cognition. But there is one thing, is that the analysis of brain network dynamics is very complex. So there is a lot of complexity that a bit hinders the advance in understanding what are the mechanisms behind the formation of these networks. And so usually there was this issue of using static connectivity or seed-based approaches, but there is an urgent need to find simplified frameworks to characterize dynamic functional connectivity at the whole brain level and to compare across conditions. So strategies to use this, understand better what is going wrong with these networks and use them and transpose them to diagnosis and the development of better therapies to rebalance these networks. So what I'm going to show here is a method that I have been using. So we developed it in 2017 already to try to characterize the formation of these brain networks at the instantaneous level and compute statistics on it in order to compare across data sets. So if we consider that this fMRI signal has an oscillatory behavior, it is possible to capture the analytic phase of the signal using the Hilbert transform. So I did this mini video just to illustrate what this means. So if we have the bold signal here over time, detected, for example, in a brain area, when we compute the Hilbert transform, is as if we create an imaginary axis. So it generates like an oscillatory signal, and this signal, and we can calculate at each time point. So each of these arrows corresponds to the phase of the bold signal at each instant of time. And this is like a collapse of this over time. So we can have the plot of the phase representation of the signal across time. Sorry. Okay. So once we have this, once we have the bold signal phase over time, we can calculate then how two different brain areas are aligned in phase. Okay, so here we have, for example, the in 1TR, this would be the bold, each arrow here represents how the bold signal is evolving, if it's going up, if it's going down, following the Hilbert transform. So here, for example, each arrow represents the bold signal phase in one brain area. But then this is still quite complex, so we still need to reduce this, or to use a way to represent this whole brain phase configuration using a simpler metrics. So what the method I've been working on is called leading eigenvector dynamics analysis, but basically it's a method to characterize the fMRI signal phase relationships at the whole brain level with reduced dimensionality. So imagine this is for a single TR, again, how the bold signal phases are organized. And what we then take is how these phases project into the main, like the leading eigenvector is represented by this vector here, how these phases project into this vector. Just even if you feel so, trying to simplify this, as John said, I want to simplify. So it's like as if we try to find which is the main orientation of bold signal phases. Imagine they're all going down, and then we capture which areas are also shifting from this main orientation. So the ones that project in the opposite direction. So this gives us a vector, which is this leading eigenvector that is here represented on the right. So for each TR, we can have a vector that says, okay, here, all these brain areas, the brain areas in blue are evolving in phase, and the ones in red are evolving out of phase with the rest. And, okay, so now if we take at each TR, so this is the vector. Here, for example, I'm coloring the brain areas whose signal is shifted from the main signal orientation. So it shows like it looks like a relevant brain pattern, but I will go into this later. So what we do is we take all these vectors at each point in time, and we can, so this is a video illustrating so the brain areas in red here that are shifted are the ones forming these sub-communities, let's say, that form this apparently noisy, but as I will show, they are quite meaningful network configurations. And then that is the idea, is to see, are these bold phase coherence patterns any meaningful for brain function? Like, do they reveal any meaningful network systems? So what we did is to use a classifier to detect recurrent patterns. Using unsupervised clustering was what so far has given us the best results. So we take all the leading eigenvectors detected across a large number of scans in a given cohort. I already tried with different data sets and everything, and we cluster this into K clusters, let's say. So one way to represent this that we used in a paper where we applied this to the Human Connectome project data from 100 subjects. So if each of these dots on the left, this would be the cloud of observations from the Human Connectome project cohort. So each of these dots represents one TR, and represents each of these dots corresponds to an eigenvector at each instant of time. And they are projected into their main principal components. So when we do a k-means partition, it's as if we are dividing this cloud into clusters. And then we can look at the cluster centroids, which means the pattern that is most representative of each cluster. And by doing this, we find, for example, here we cluster into 5 states. And we find that the main state of bold coherence, let's say, bold phase coherence, is when all bold signals are going together in phase. So there are some points in time where the brain activity is characterized by a global mode. And this is most of the time. Some people have discussed, and maybe if you have done similar analysis on both activity, there is always this issue of the global signal, if it should be removed or not. We tried removing the global signal. This global state disappears, but then the algorithm tries to detect a pattern the patterns that come out appear much more random, because it appears that this global signal is actually meaningful. And if we remove it, we get noise, basically, during those time points. But what is perhaps more interesting is that the other patterns that we detect overlap with known functional subsystems. So exactly. So here I overlap with known resting state networks from the literature. So on the bottom, we see reference functional network systems from Thomas Yeo, 2011. And so they divided the brain into the visual, somatomotor, dorsal attention, limbic network, frontoparietal, and default. And we find that the network that appears with second most probability of occurrence, which is this second state, has a strong overlap only with the default mode network. And if we look at the third pattern of, let's say, bold phase locking, we call it here PL, stands for phase locking. We find the frontoparietal system. What does this mean? Is that during these TRs, the TRs that the time points across all the subjects that were assigned to this state, the signal in the areas corresponding to the frontoparietal system is going in one direction, and the rest of the brain is going in an opposite direction. What I've been having insights now is that this suggests there is the existence of standing wave patterns in the bold signal that is driving the generation of functional networks. So for example, here, if we look at, here is the visual system, and we find that, when the signal in the visual system goes up, the rest of the brain goes down. So there will be it's, you know, when you see a standing wave in a drum or something, vibration, we see that some areas can go up while the others go down. So there seems to be a standing wave property in brain activity. But anyway, apart from that, so here we find one of the networks here overlaps with two known reference networks, but that's also because here we chose just k = 5. So if we split into two states, we would get only the global and the DMN. If we split into 3 states or to 4 states, as we increase the number of states, we start seeing more and more networks coming out. Especially here in this data set, since the Yeo network didn't cover subcortical structures, but we find from k = 7 a consistent subcortical system. From k = 9, we find consistently the limbic system as well as the third most prominent state. So it's interesting to find this overlap. We also calculated the silhouette value to see how well the system can be described by this partition. And we find indeed that in the brain, there may be, like, at least at this macroscopic level, using 3, 4, 5, 6 network states is optimal to represent whole brain activity at the large scale. Of course, then what is interesting is that as we increase K, we find more fine-grained network configuration. So this is a bit an overview of the approach, the methodological approach, where we have the signal in each brain area, we compute the eigenvector each instant of time, then we cluster them and we detect these recurrent phase locking states. Now, in theoretical or in conceptual terms, what this helps us to understand is that functional network states, in a way, can be approached as a trajectory in state space. So imagine if each of these crosses represents the centroid of a brain state. I don't know if you can see it over time, but it's like the brain is constantly exploring different possibilities. So it's constantly evolving over a state space landscape. So we can characterize brain activity as a trajectory in this state space. So it's a conceptual framework, let's say. And then the idea is this dynamical exploration of these network patterns relevant for psychophysiology? So to address this question, we need to define single scan metrics to compare across conditions. Now, this approach to assign each time point to a given bold-phase locking state allows us to define measures such as the state probability. So FC state is functional connectivity state, is the same as phase locking state probability. So we can count the number of time points that are assigned to a given cluster in a given scan. Well, we can define these measures for each single scan. It's one of the things that I will go into detail on the different measures. So one of them is the fractional occupancy, is how often your brain spends in a specific phase locking configuration. So imagine how often you spend close to the default mode network. And we define this measure. It's like counting the number of blue bars here. Another measure is the dwell time, is to calculate the number of consecutive time points where you enter a specific state, how long you remain there. So this from dynamical systems theory, in this abstract framework of brain activity, wandering around these network states, it gives us insights of the trajectories of the dwell time, is like the attractive force of these states. And another measure we can use is to characterize, so here is not the dwell time, it's the Markov chain, but I have an illustration here. Okay, so to also from dynamical systems, something we can use is to calculate the transition probability matrix. So if you know that you are in state 1, for example, in the global state, what is the probability to remain in that state? So here we find that when people are in the global state, there's a probability of 77% to remain there. But then with 8% probability, they switch to the default mode network. And then it returns to the global system with higher probability than switching to the others. Okay, so this is what we get from this type of matrices. So here we find that when we are in any of the states, there's always higher probability to remain there or to switch back to state 1. So from this type of matrix, we can have an insight that the global state is like a baseline activity state of global oscillation, very slow at the scale of the fMRI signal. And then these other states that appear sporadically and are short-lived, and they have less stability, let's say, these can be termed ghost attractors. Ghost attractors or metastable attractors or states that when you can switch to those states, but then immediately you return to baseline. So this is the methodological framework, let's say. So we would have this, for each fMRI scan, we can calculate the occupancy, the dwell time, and this transition graph. And now I'm going to show some results on how to compare this across conditions. So here, this is the second part of the talk. So all of these studies have been done, so I developed the method, and then the idea was to start applying it to existing data sets. So I didn't design my own experiments, but I applied the method to existing data sets. Because as you know, there's an issue now on the neuroimaging community that there are a lot of data sets. They were tried, sometimes they were analyzed with standard techniques. And now what we really need is new methods to analyze the data rather than just having new data sets. So we analyzed data of people with cognitive decline, major depressive disorder, psilocybin. I will go a bit into detail on this. So one of them was to compare healthy elders. So they had they were older than 50 years old. And they were compared, they were scored, let's say, according to their level of cognitive performance. So they did a bunch of tests, like counting backwards, jumping every tree, you know, these type of things. And so they chose the best performers and the worst performers, and these were invited to be scanned. And so I had to analyze this. So there were changes that had been detected already at the level of white matter and also at the level of static functional connectivity, but the results weren't very insightful into what is driving better cognitive performance. So the idea was to first apply this LIDA method. So here, this is still 4 years ago, so the renderings of the networks was still very preliminary, let's say. But what we find is that there are these different phase locking patterns. Again, the global state, this was the limbic network, the frontoparietal merged together with the DMN. And we found these patterns in the brain activity of all participants. But then when we look at the probability of occurrence, so after we run the k-means, we compare the probability of occurrence of these states across subjects. And we found that there was a significant difference between the occupancy and the duration of these networks between groups. So this gives us an insight that maybe cognitive performance is related to this dynamical exploration of brain networks during rest. So another thing that we found is that there was changes in the switching probabilities. So there were significant changes, like the good performers had more probability to switch from some states to others, like to switch back and forth to the DMN compared to poor performers. Poor performers exhibited more time in networks that are more structurally shaped. So there were some insights that revealed something was going on. So then we tried with another data set. These were subjects that had major depressive disorder, MDD. And they were now in remission. They were not taking any medication. And the idea was to compare these people with controls, to see if there was difference in their brain activity. So again, we ran the same state, then the same analysis, the same algorithms, and we detected, well, this is complicated because it had different tasks and things, but what we detected essentially is this network number 4 is where we found more significant differences, both in terms of probability and duration, that occurred significantly less in patients. So this is the plot. So this network here, it includes areas of the DMN and frontal areas, but also salient and subcortical areas, including the thalamus and dorsal striatum. So this is quite relevant for psychiatry because these areas are known to be involved in depression. And in a way, it shows that those people, they are at risk of having major depressive disorder again. They have like a failure to engage or to form this specific network pattern that apparently may be relevant for psychiatry, let's say. And also so here, this is again looking at the trajectories, and we find that the controls, when they enter this network state, that already is less probable in patients, but when either the controls or the patients enter in this network state, the controls will switch from here more often, significantly more often, to a network comprising the connectivity between the prefrontal areas and the striatum. And this is very relevant for psychiatry because the striatum is known to regulate mood in a way, and the prefrontal cortex is what, in a way, orders or processes this information. So in a way, it would be that people with MDD fail to regulate their mood. So this was quite nice because the results came out quite meaningful. This was one of the strong points of this, because it could have given something totally random. And then we tried another data set, and this one was nice because it studied the effect of a psychoactive compound. It's a psychedelic. So this was a collaboration with the Center for Psychedelics Research, where they gave us the data set where people, so controls, healthy people, were infused inside the scanner without moving. After 5 minutes of scan, they were infused either with psilocybin, which is this psychoactive compound that is found in magic mushrooms, and then they were 2 weeks apart or over 1 week apart. They went back again and they were infused with the placebo infusion. Okay, and then we did again this LIDA analysis to see which network states were being activated at each point. So this only one of the 4 scans. So if each of these things represents for 1 scan, right? For each subject, we have 4 scans where only 1 is psychedelic. Okay. And so first we detected again like this network states that we detect with LIDA. So the global state, then one that overlapped strongly with the default mode network. This one overlapped very strongly, the number 3. So this is what I show at the bottom plot. The blue bar, the light blue bar, very high, means that our state number 3 from the top overlaps almost 80% or 75% with the frontoparietal system. And then the others a bit weakly to the limbic system, but still significant. So that there is this overlap with, again, these reference systems, so that just to validate. And then, okay, we compare their probability of occurrence. So here I am showing at the bottom, the bars represent the probability of occurrence of each of these states in the placebo condition. So we see no difference. So before and after the placebo, there are no significant differences between conditions. Now, before injecting the psilocybin, the probability of occurrence was also not revealing any significant difference compared to the placebo. So, which means, okay, there's some reliability in the probability of occurrence of these states. Now, when we consider the psilocybin session, what we found is that only two patterns were affected. And these patterns were affected because when we injected psilocybin, there was a sharp decrease in the probability of the frontoparietal system, which is associated to executive function. And that was and once we cleared out this kind of ghost pattern that would be formed, there is a concomitant increase in the global baseline activity. So it aligns with this conceptual framework that we would have this baseline state and then we would be switching to these patterns. And in a way, this is nice because it shows that there is a molecule that can selectively modulate the occurrence of a given network system, which is nice because it may relate to, so in a way, okay, so this is the cloud of states. So before injection, both placebo and psilocybin, so this would be the cloud of states with each color representing the occurrence of a given pattern. And then when we inject either this red or this blue compound, here we see the cloud again. And now this is the question to the audience. You don't need to reply, but the idea is to find, just by looking at this cloud of observations, like, can we visually see some difference, right? So here we see that the amount of dots of trajectories, like over time, it significantly goes less to this red part of the cloud, right? So after this would be the placebo. Let me see. So this is the question: which one of these would be psilocybin? So psilocybin would be here, and it's like it, in terms of trajectories, it's like psilocybin disrupts all the trajectories to that functional network state. So there is some relevance, and here is also it's very interesting because each subject scored its psychedelic experience, let's say. And so we found that the occupancy of state number 3, like something like an objective measure that we can get at a single scan level without asking anything to the person, strongly correlates with the subjective experience of the person. And it's not just okay, there's more complexity or there's more entropy. It's a specific network configuration that correlates to the subjective score. So it's interesting. And now we got even more strong and exciting results with the DMT. So the other paper was published, and now we got a new unique data set from people with DMT. So as you see, the renderings of the networks are starting to improve. So in a way, the global mode would be, now it's represented like this. But so we find in all non-psychedelic conditions, there is a consistency across subjects on the occupancy of these states, okay? And then the fourth column in this case is the DMT, is after DMT injection. The paradigm is exactly the same as from the psilocybin. And here with DMT, it's not just the frontoparietal, although this is the strongest effect. So it goes 10 to minus 8, the p-value. It's much more significant than what we had in the psilocybin study. But we also find a decrease in the occurrence of the default mode network, which links to the literature of DMT, and an increase in the limbic state. So you see here. So it's nice because it kind of keeps validating the approach of this strategy. Also here, maybe people may be wondering, what about corrections, statistics, multiple comparisons? So here in the DMT, we are really using a Bonferroni threshold. So what this means, so this is if I compare the occupancy between before and after placebo, and this is the p-value. So the red line here corresponds to the 0.05 threshold on p-values, like compare pre versus post placebo. This is the number of states that we consider. We can choose 5, 6, 7, 8, 9, 10, but we see that irrespective of the number of states that we choose, nothing falls below this blue line, which is a Bonferroni threshold that corrects for all the comparisons that we do. Now, if we compare again these non-psychedelic conditions with each other, none of them again passes the blue threshold. So these red dots here that pass the 0.05 threshold are these false positives that arise when we compare many things together. Now, when we compare with the DMT, so these are comparing pre-placebo with post-DMT, or post-placebo with post-DMT, or pre-DMT versus post-DMT, all of them show for any number of clusters that we choose a significant difference. So in a way, any measure that we use to compare pre versus post DMT will give us differences, because indeed the activity seems much more, visually there is a difference. But now the advantage of this approach is that it can characterize and identify exactly which are the network systems that are going up and which are the ones that are going down. So I think it can give us insights into this psychophysiology. Another data set interesting that I'm also finding very relevant is this data set with people with obsessive compulsive disorder. So when we compare people with OCD with controls, most of the networks have consistent, so we don't detect differences, and there's this state that connects subcortical structures with, I will show a plot, that is significantly suppressed in people with OCD. And this network, if we look at the list of brain areas, this is the anterior cingulate, and then all these areas are like the caudate, putamen, pallidum, and thalamus. So this is the striatum and thalamic system. And this connection of to the anterior cingulate is really aligned with what people what neurologists and psychiatrists have thought of as a model for these disorders, these psychiatric disorders. So a failure in the regulation of these mood regulators that are the subcortical structures that regulate the mood. So it's nice, again, it seems to be revealing some interesting things. Another nice study that we've been doing, this is collaboration with UCL, is with people using deep brain stimulation. So one of the things in this project is that people who do deep brain stimulation have a tendency to report that they feel better. So it's not that they just reduce the tremor, you know, it's used to cure Parkinson's disease, not to alleviate the symptoms of Parkinson's disease. But some people where the DBS sometimes it's not very well placed and it doesn't relieve the tremor, but people report that they feel better with the deep brain stimulation. And so now deep brain stimulation is also used for psychiatric disorders, for mood disorders, because in a way it seems to be affecting this limbic system. And what we found, so this is the patterns, so here is a rendering in 3D, but they are colored in white because they overlap with the limbic system, but especially here on the left panel. So this is when DBS is turned off. There's a lower probability. When DBS is turned on, there is a significant increase in the probability of occurrence of this system here, that in the AL parcellation, it's called the rectus and the olfactory bulb, but it's also the orbitofrontal cortex that relates to reward and to regulation. So in a way, stimulation in the subthalamic nucleus, but probably in the more frontal parts, projects into this mood regulator system that helps people feel better. And of course, we find a significant decrease in the somatomotor system, linking to the alleviation of effects. And this is children listening to music. Also, we find that they activate this orbitofrontal reward system as well using mood. This is another one where we published using LIDA, but on a data set from women watching babies that were smiling more often. Well, this is the paradigm, but what I want here to show is that there is a pattern. So we identified this pattern that relates to the happiness score of the babies that were presented. And this was published in cerebral cortex because the network that was detected is highly meaningful. So it really relates the orbitofrontal cortex with the insula, the hippocampus, and the amygdala that are known or have been hypothesized to regulate this learned infant emotionality, let's say. So it was interesting. Then there's another one on self-reflectiveness that increased the occupancy of the default mode network. And overall, so as I want to recap a bit, what I'm finding is that this occupancy of functional networks can have, like, we can kind of think maybe in the future of how this could be used as a potential biomarker, or just to see that at least this is making sense, is that there seem to be reference values for healthy people without any type of drug or medication. Then we find some moderate reliability. So we also found that people scanned twice on the same day have reliability on the measures. So the probability of being in the DMN is quite consistent. Then we are finding altered values in some psychiatric disorders, let's say psychopathology. So in OCD, in major depressive disorder. We also did a master project in schizophrenia and autism. And also with these non-clinical symptoms, so this is people who really have a psychiatric disorder, but it could be non-clinical symptoms like just the capacity to think about yourself, like self-reflectiveness, or also we published one where the people were suffering from a broken heart, like a relationship breakup. And we found also a relationship with their like altered values when people were sad, which is a trigger for depression, but it's not considered a clinical thing. And then, well, there was something wrong with these bars, but the idea is we are finding as well that we can tune these values. So we can tune the occupancy of these networks using either psychoactive drugs, also brain stimulation, and natural stimuli like music, or sorry, this is moving around on its own, like natural stimuli. So what I want here to recap with this thing is that there seems to be important information, you understand, in the dynamics of these networks, or how much they prevail over time, that can help us better understand brain function. So overall, what can we learn from the results or from this kind of approach is that if we merge these data-driven findings with the theory or the physics of dynamical systems, we find that the relative stability, the probability of occurrence is a bit the attractive force of these patterns, relates to brain function. So it is modulated by drugs, neurotransmitters, or by electromagnetic stimulation, but also by training, by psychotherapy. So it could be nice if we could modulate this landscape or these trajectories even naturally, so not just with artificial networks. So quantifying these dynamics, I think, is a way to go to provide better insights to improve in the diagnosis and treatment of these disorders, and also for the models, because I think once we have a better insight from the data, we can design better models, and I'm on that as well. So, but so I did this slide for a different thing, but for a pitch. But ultimately, what this would lead is that if we could help psychiatrists not just the question and answers that they typically ask, but if they ask to quantify, right? Okay, someone comes to the psychiatrist with some symptoms, and the psychiatrist can prescribe a report. So the person would go to a 30-minute resting state scan. This data could be sent, to some cloud computing system that would generate, run the algorithm, compare with reference values, and deliver a report. And more, if we have computational models that can help us understand how to modulate the occupancy of these networks, the system we can use AI to provide decision information to the psychiatrist, to say, okay, we think based on these large like computational models and data sets, that if you give this specific drug, it will help rebalance this activity. So then the doctor can have an assisted decision making, let's say, and increase the chances of helping the subject. So this would be more like an implementation. And here I'm on work in progress, so improving this algorithm. I've trained different people, like trying to use this, but now I'm working on trying to put this as a platform with tutorials, with code of conduct and that, and also put all the codes as a software package on GitHub to help. So this is a bit it was a side project that came out while exploring really what is going on in brain activity. And that's it. So I would like to acknowledge, so I have my group in Portugal, but I'm still very closely linked to the lab where I did my latest postdoc in the UK, and with Gustavo Deco in Barcelona, where I did my PhD. So this is a picture when they all came to Portugal 2 years ago. And also in Lisbon, I'm also in close collaboration with a lab in Lisbon. And well, thank you all for your attention. I hope I'm kind of on time. Thank you for all the, thank you.
Thanks a lot, Joana. Splendid talk. Yeah, a lot of ground covered there. You've done so much work with this methodology, and really, doing something that I think there's a lot of room for more of, which is taking a tool, taking a technique and really trying to understand it intensely across a lot of data sets, and means that you understand the method well, but you also understand the things that you're seeing in the questions that you're asking better by applying it across a wide set of diverse set of data sets and research areas. Yeah, really great. So we have some questions to go. So I'm gonna just read off some cues that we had from some other people, and I have one or two for myself. Just a reminder, everyone else, we've got time for more cues if you want to just pop them in the chat, and we'll get Joana's thoughts on the. So number one, first one's coming from Itai Hai, and Itai is asking, what are the time scales of the pattern switching typically?
Okay, hi. So that is actually a good question, because it will depend a lot. Like the smoothing and on the data that we have. So what we have so far is fMRI signals with TRs that can go as slow as fast, let's say, or as short as 720 milliseconds, so a little less than a second in the Human Connectome project. This is the shortest TR that I analyzed so far. And what I found was that before we were smoothing the fMRI signal below 0.1 hertz. So by definition, by smoothing it at these slow scales, the states seem to be lasting like 10 seconds, 15 seconds. But then if we didn't smooth, and if we allow so includes all the frequencies up to the Nyquist frequency, which in the HCP data set was up to 0.6 hertz, we found increased reliability. So within subject reliability by including all the frequency components, and we found that the dwell times can be as low as 1 TR. So in my intuition and what I thought from this analysis, so we published this in 2020, and now what I'm looking at, maybe to relate this to MEG data and all these things that report that these states are occurring at the 200 millisecond scale, probably. So when we see these kind of slow patterns forming in brain activity, can be a consequence of the low pass filtering we are doing. And maybe these configurations are really short-lived. So that's the idea of the ghost thing or something like, it's just like sporadic excursions that you would go and come back. So, but we would need faster acquisition sequences. And this is what I'm doing in the collaboration with the lab in Lisbon. So we are doing on rats, and we reached the single slide acquisition because I just wanted to see, okay, how fast is this switching? And we reached the 38 millisecond TR, which is, and it's giving insights. So it's not just that these states activate and deactivate, is that these states resonate, which is even interesting. So the activation of a given resting state network is kind of a resonance. And so when we are looking at co-activation patterns or these leading eigenvectors, we are just capturing when it's going up, but it seems that the pattern, well, but this is unpublished insights. But yes, the dwell time and the duration, we can't, so to reply your answer, we can't give an absolute answer on that with the timescales of fMRI acquisitions of the current ones.
So that's interesting. So you're doing scanning and then the acquisition time down to, you said 38 milliseconds.
Yes.
That's wow. They can do crazy stuff with those animal scanners.
Should be coming out soon.
Right. Look out for that. But this leads into Itai's question. Actually, leads into one of the questions that I had, because another area that you've spent a lot of time on with your collaborators in Oxford and elsewhere is fast time scale activity and like network structure in EEG and MEG, right? So and those guys in Oxford, they've been doing these hidden Markov models, and like, you know, there's a lot of you know, synthesis between the approach that you've had here. So how do you think about that, a kind of, you know, variation on Itai's question, that, you know, there's state switching going on at the millisecond level, at the tens of millisecond level. You're kind of a little bit over to the right in the, you know, the seconds level, tens of second, but there has to be a continuum here. And have you thought about how, you know, to interpret the patterns you're seeing in fMRI with respect to the things that you've also seen in EEG and MEG? Is there a convergent picture, do you think?
I hope there will be. I am putting that as my motto for understanding brain function, is to link the findings we are detecting with electrophysiology with what we detect in fMRI. We are on the way. Indeed, I've discussed a lot with Diego Vidal with that, why we are, what is the difference between the hidden Markov models and the phase locking patterns. What I'm finding is that the hidden Markov models assume that, so the activation of a system would be just, it activates and deactivates. And with the LiDAR, we kind of allow so the activation can be either a co-activation or a co-deactivation. I don't know if I make myself clear, but we are capturing like a pattern that can be rotating over time. So in a way, we would be more sensitive to resonance. But ultimately, yes, I think we are just looking at the same thing from slightly different angles. And the thing here is to keep fine-tuning, and you know, I'm open to integrate methods from other groups as I see if they work better. But indeed, I mean, ultimately is to improve the temporal resolution of fMRI may help, and maybe improve the reconstruction algorithms from MEG. But yeah, I think a model in the end will be the answer to combine these two fields.
So I've got another thing that follows on from there. Another question is, you know, you've been looking at these phase patterns, and I mean, there's maybe three key principal ways that people analyze functional connectivity in fMRI. They look at Pearson correlations, they look at ICA components, and they look at phase patterns of correlations in phases, which is what you've been doing. But between like in a lot of the leader methodology, this network eigenvector thing, you could do that with Pearson correlations. So and I guess you have, but I'm just wondering if you could kind of comment on what's
the difference,
why you're taking it that way, and is there any kind of major significance
with the correlation is that
taken?
Yeah, of course, I'm taking I think I know where you're reaching. So the thing with the correlation was that there was a debate on what was the best window size, right? So we wanted to get rid of the window size, and by looking at phase locking, we can look at the instantaneous level. So in a way, if I average all the instantaneous phase coherence matrix, I get something that correlates 99% with the correlation matrix, the static. Or if I divide into minute-long windows and I compute the correlation matrix, it's the same. But the correlation, in a way, you could compute the correlation instantaneously. It's like the outer product. But here as well, it's not just the correlation between the signals, it's correlation between the Hilbert transform, and the Hilbert transform has something that even if you see a zero, like in the bold, you see like it would be crossing a zero, you don't know if this zero means that the bold signal is going up or it's going down. And with the Hilbert, we also have that information. So in a way, it's complex to describe, maybe with the further conversations, but in a way, I think what it's capturing is it's more sensitive to these standing wave effects that, in a way, correlation also captures synchronization, okay? But it also captures, let's say, aperiodic co-fluctuations, which is one thing. Aperiodic co-fluctuations, they would give a correlation matrix, or if it's stochastic resonance, let's say, so these patterns resonate and then go back, they would also give the same correlation matrix. So here it's a bit to disambiguate what is going on. Are these aperiodic coactivations or kind of stochastic resonances, like something that is going up and down? And I'm on that direction now to disambiguate these two concepts, but it's not trivial.
That's really interesting. Okay, so we have a question from David about, so David says thanks for the talk, and he's wondering what your thoughts are on the role of structural connectivity. For example, do you tend to have more jumps, more transitions between areas that are highly physically connected?
Okay, so that is a very nice question, and it brings us to the models, right? So what we find from what I can find in very theoretical works on network dynamics, so let's say if you have a connectivity matrix, even if it's not the brain, just a graph of a network, if this system has specific topological features, let's say cluster or community structure, okay, the system can be in a regime where only like one cluster synchronizes, tries to synchronize, let's say, but then it desynchronizes, and then another cluster synchronizes. So there's a kind of competition between clusters. And there is, for example, one paper on called metastable chimera states in coupled oscillator systems, for example. And what this means is that you can, even without noise, a specific network configuration can have a self-sustained, a stationary activity, like just spontaneous activity that is constantly showing like the activation or the resonance of different subsystems. And so in a way, of course, yes, the structural connectivity has a huge role on this. But also there are parameters in the model that we can tune to have more or less of this activity. So I believe, indeed, yes. So ultimately, everything is shaped by the structure. So we have to understand better how to tune this structure. But I also believe that, so you could have the same structure, but then you just take a drug, you know, and your dynamics is different. So it means that also it doesn't depend only on the structure. So it's a combination of the two things.
Right. Okay, so we've got two kind of methodological-ish questions come to, I guess, well, we've got three. Let's see if we can get through these two, and then maybe we'll wrap up the last one. So we've got a question from Neda Rashidi, which was, I'll just ask these two together. She's saying, is there a minimum time needed for the fMRI data? Does it need to be 5 minutes, or does it need to be longer for kind of reliability? And then the other one is from Dan Muller, and he's asking, have you looked for response to treatment phenotypes in any of the clinical studies that you've done?
So two very interesting questions. So the 5-minute clinical data, what I can tell is the psilocybin data set, just to give you an insight, we had only 9 subjects. Each scan was just 5 minutes, and the TR was 3 seconds. So we only had 100 time points per scan. Okay, and only 9 subjects. So at one point, it was like the weakness of the data set, right? We were like, no, we will never publish anything with this. But on the other hand, it was a strength because we ran separately the things on the placebo data and on the psilocybin scans. So we separated. So it was really validated because it was impossible. Like if it was just a random result, how could the scan like that was processed afterwards 2 weeks later have the same probability as in the others? So it was a way, it was also a strength of the method to show that it worked in such a poor, or not poor data set, but well, weakly sampled data set, like you said. So this was TR of 3 seconds, 100 points per scan. The other question regarding therapy. I just received this week data set, just downloaded 100 gigabytes from people with obsessive compulsive disorder who did cognitive behavioral therapy, but I haven't analyzed it. So the idea, we have controls and patients who did CBT. And the idea is to see, okay, if the differences that we detect between controls and patients, of course, if these differences are minimized after deep CBT, but I haven't opened it yet. So, but it's on the to-do list.
Great. Okay, we've got one more methodological question from Marcos, and I want to get this in because he asked first. So but I kind of didn't get to it. So he just wanted to know: is the k-means analysis conducted in each data set separately, or are the clusters found in the first data set and then predicted across the other ones?
Interesting question, and it also aligns with, but yes. So one of the issues that we've been trying like to reinforce the validity of the method is we run the k-means on half of the data set. For example, in the Human Connectome project data, we did it. So each person was scanned twice. Okay, so we have 100 scans from the first session and 100 from the second. So we determined the k-means from the first 100, okay? And then we just used those centroids, okay? And just projected it on the others. So the fact that the probabilities are reliable are a validation, because the centroids were not taken from the second scans, the second data set. What I've tried now was to use the HCP centroids to the DMT data set, okay? Or for example, in the deep brain stimulation, the controls are the ones from the paper of the healthy older adults, the ones from cognitive performance. So it's the same centroids can be found and with reliable occupancies and that probabilities from different labs. Right now I'm on 28 data sets and 2 of them gave nothing, but it happens. I don't know if it's the problem of the method or of the data set. Also, some pre-processing, we are finding some pre-processing steps are critical, like the removal of the global signal, but also this ICA aroma removal of components. Some of the things then when we really compare, so we went back to all the pre-processing steps, and we found that some of them were really creating artifacts in the data. But yeah, there is some consistency. That's what I can reply.
That speaks to the issue of heterogeneity in pre-processing. I kind of wonder
exactly.
how much imaging results are it's just so hard to compare across studies sometimes.
It's less there. There's more, people are more. So what drives more variability between studies is not the variability between subjects, is more the variability between pre-processing.
Right. Yeah. That's bad. Well, I'm glad you've been paying close attention to that.
Yes, well, because but I'm finding some actually the less you clean the data, the best. No smoothing, nothing.
Okay, well, so thank you very much for the fascinating talk, Joana. Very impressive work again. Like, I think the approach of taking a hammer, sculpting the hammer, refining the hammer, and then finding a lot of nails, like there's a lot to that, and you've really managed to amass a lot of impressive results with this approach. Definitely interested in talking to you.
I used to say we found a field that will keep us busy for the next 50 years. So
yeah, it keeps on giving in neuroimaging. And the standing waves ideas are really interesting, and interested in kind of seeing where you go with that, and discussing with you more on that in the future.
Yeah, hopefully in some conference.
Yeah,
physical meeting.
Soon. So just thanks again on behalf of the KCNI. Thanks to Joana Cabral, and bye everyone. Have a good day.
Bye.
Thanks, Joana.
Bye.